So now the next thing is that how did we go for naming the restriction enzymes? So there is a universal system for going for nomenclature or naming of restriction enzymes. So the first thing you know that what is the function of restriction endonuclease? The function of restriction endonuclease, it depends upon the specific DNA nucleotide sequences. So how does restriction endonuclease know that where have I to go and cause the cut in that long length DNA molecule? This is a cause of concern for restriction endonuclease, how to go for hunting for a site where to put the cut. So the first restriction endonuclease isolated was discovered was EcoR1, but the first to be isolated was HIND2. Okay, this HIND2 restriction enzyme, what does it do? It always cuts the DNA at a particular point. There is a particular point in the DNA where it puts the cut. Now what is that particular point known as? That particular point where the restriction endonuclease recognizes and puts the cut, there is some specific sequence present there. That specific sequence is very special. It is a sequence which is read from front to back and from back to front. That is from 5' to 3' direction on one of the threads, and on the other strand of DNA also it is read from 5' to 3' and similarly. Say for example, here in the slide you can see EcoR1 is the restriction endonuclease which was isolated from the first time from Escherichia coli. You can see its recognition sequence or palindromic sequence. This is the sequence which is read similar from both the directions. Okay, so you see what is the palindromic sequence here? It is GAATTC. And you see the other strand. When you read it from 5' to 3' direction, it reads similar. It reads same. That is GAATTC here also. In the same way, you can see the next example. It is TAC1. What is TAC1? It is a restriction enzyme which was for the first time isolated from a bacteria, Thermus aquaticus. This enzyme again, it also has got a palindrome. What is its palindromic sequence? A sequence which TAC1 recognizes is TCGA. If you read it on the other strand from back to front, that is 5' to 3' side, you will again get the same reading pattern, that is TCGA. Okay, so this is how does the restriction endonuclease actually function? It functions in a way that it goes and recognizes its peculiar palindromic sequence. What is that palindromic sequence? That the enzyme knows. It goes and finds out that sequence, and then it recognizes a particular site in that palindrome where it has to make the cut. Say for example, here again you see in EcoR1, if the palindrome is GAATTC, then the recognition site for EcoR1 is between G and A. So the EcoR1, it puts the cut between G and A on both the strands. So this is how the restriction endonucleases they act. Okay, so there are altogether almost 900 different types of restriction endonucleases being isolated. These are isolated from 230 strains of bacteria. Okay, so now let us start with the nomenclature, the rule for nomenclature of restriction endonuclease. So let us see. While naming restriction endonuclease, the first letter of the name, it comes from the genus of the organism from which it is isolated. Say for example, here on the slide you can see Hind2. Hind2 is the restriction endonuclease. Here, Hind2 is isolated from a bacteria known as Haemophilus. So the first letter H, it comes from the name of genus of the bacteria, that is Haemophilus. Okay, and the second two letters, that is I and N, it comes from the name of the species of the source bacteria from which it is isolated. So IN stands for influenzae here. So it means this restriction endonuclease Hind2 is isolated from bacteria Haemophilus influenzae. So H comes from Haemophilus and IN comes from influenzae. Okay, the fourth letter, in fact, it is derived from the strain of organism. Which is the strain of this organism? Okay, so this D, it is derived from the particular strain of Haemophilus influenzae. And then you will find in the name there are certain Roman numerals, that is 2 you can see. These Roman numerals actually, they depict the sequence of discovery of this restriction endonuclease. So I hope you would have understood. It is how do the restriction endonucleases get their names from? They get the name from the name of the genus, name of the species of the organism from which they are isolated. The fourth letter comes from the strain of the organism, and the Roman numerals, they are derived from the sequence of discovery of the restriction enzyme. So just here I have just taken down a few restriction endonucleases to show you as to how they have got their names. So let us see the first letter of the first restriction endonuclease, that is EcoR1, comes from its genus, which is Escherichia. Second and third letters, they come from its species, that is coli. The fourth letter, that is strain R. R comes from the strain of this bacteria, and the sequence of discovery is, this is the first restriction endonuclease. That's why it's known as EcoR1. For Hind2, just now I explained you in the previous slide, and let us again see now the third restriction endonuclease given here, which is BAMH1. What is BAMH1? BAMH1 is the restriction endonuclease which was isolated for the first time from a bacteria known as Bacillus amyloliquefaciens. So this is the bacteria. That's how it has got its name also from. The first letter BAM of BAM, it comes from Bacillus. The second two letters, they come from amyloliquefaciens. Then the fourth letter H, it is derived from the strain of the bacteria, and the fifth Roman numeral, it is actually the sequence of discovery of this restriction enzyme. So now let us see how does the restriction endonuclease act. Although I have told you before, but let me repeat myself here. The restriction enzyme, what does it do? It inspects the entire length of DNA sequence. You know that DNA is a polymer made up of nucleotides. So it is actually a huge, large molecule. So this large molecule, the restriction endonuclease, what does it do? It goes around. It just moves and it tries to locate that peculiar sequence which is known as palindromic sequence. And I already told you what is a palindromic sequence. Palindromic sequence is a sequence of nucleotides which are read in 5-3- direction, similar on both the strands. Okay, say for example, here you can see on the slide, I have written the palindromic sequence of ECO-R1. It is GAATTC. And on the other strand also, if you read it from 5-3- direction, again you will get the same sequence GAATTC. So this is actually the palindromic sequence. As soon as ECO-R1 molecular scissor is used to cut the DNA, the ECO-R1 goes, inspects the entire length of DNA which it has to cut, and wherever it locates this palindrome, it goes there and it spots the site where it has to put the cut, and that site is known as the recognition site. So let me tell you, here you can see those red arrows. What are those red arrows? The red arrows in this palindromic sequence are actually the recognition sites. So what does this restriction endonuclease ECO-R1 do? It goes and puts the cut between G and A on both the strands, and this is how it is able to cut the DNA. Okay, now let's see here. When the restriction endonuclease, it cuts, there are two ways how can a restriction endonuclease insert a cut in the DNA. So let me tell you, the first type of cut which is done by the restriction endonuclease in the DNA is, I am explaining you here, just see it. The restriction endonuclease cuts the strand little away from the center of the palindrome. Okay, there are two ways. Either the restriction endonuclease, it goes and cuts the DNA right from the center. Okay, otherwise there is one other way also, that it can go and insert the cut or introduce the cut on little shifted position from the center, not right in the center. Okay, so when the restriction endonuclease cuts the strand little away from the center of the palindromic sequence, but between the same two bases on the opposite strands, this leads to formation of a single stranded portion. Those single stranded portions are known as sticky ends. Now to explain here, I have put an example. You see here again, we are using ECO-R1 and its palindrome. What is its palindrome? GAATTC, GAATTC. So once we have inserted ECO-R1, ECO-R1 will again go inspect the DNA, cut the DNA between G and A, and you see where G and A are located. They are not located right in the center of palindrome. Rather, they are located on the shifted position away from the center. So once this cut has happened, you see the two pieces which are generated by the cut. The two pieces have the hangover position. They have got a few nucleotides which are free, which are not having complementary pairs. Okay, so these are known as overhanging ends or sticky ends. So what is special about these sticky ends? These sticky ends, once they are produced, they, just because they don't have their complementary bases, they are always eager and keen to go and look for their complementary bases, and wherever they get it, no, they go for complementary base pairing, and hence it becomes very easy for the ligase to cause the joining of such sticky ends during recombinant DNA technology. So now let us see the other type of restriction endonuclease which produces a cut right in the center of the palindromic sequence. So just see here, there is a sequence which is actually recognized by SMA1. This is the restriction endonuclease. This restriction endonuclease, its palindromic sequence is CCC-GGG. CCC-GGG. And what does it do? Its recognition site is between C and G. So what does it do? It goes and cuts the DNA right in the center of the palindrome. Hence, it produces blunt ends. You don't see the overhanging sequences here. So such type of cuts which are done by restriction endonucleases, they produce blunt ends. But if you compare the two, the sticky ends and blunt ends, it is always better to go for producing sticky ends by using such restriction endonucleases which have a capacity to produce sticky ends. Now, this is a slide which is showing the different examples of different restriction endonuclease and their mechanism of action. So to see the first one, Escherichia coli, I've already explained you too many times, and you can see here the overhanging sequences or the sticky ends which are produced by EcoR1. Okay, the next one is PST1. PST1 is the restriction endonuclease which is isolated from Providentia stuarti. So this bacteria, what does it do? It again has got a palindromic sequence and it cuts the DNA between G and A, which again produces an overhang or a sticky end. Okay? In the same way, SMA, which we had already discussed before in the previous slide, that its palindromic sequences CCC, GGG, CCC, GGG, and the recognition site is between C and G. So what happens? The restriction endonuclease, it comes and it cuts the DNA between C and G, which lies right in the center of the palindrome. Hence, we are able to produce blunt ends. In the same way, there is yet another restriction endonuclease known as HAE or He3. This bacteria, the bacteria which yields this restriction enzyme is known as Haemophilus aegyptius. This again has got the palindromic sequence GGCC, GGCC, and the restriction endonuclease, it again cleaves the DNA between G and C, and just because it cleaves the DNA right in the center of the palindrome, that's why it again produces the blunt ends. And the last one is the Haemophilus parainfluenzae. This restriction endonuclease recognizes the palindrome, which is CC, GG, GG, CC, and it cleaves the DNA not right in the center, rather towards the ends, that is between C and C. So that is why again this restriction endonuclease, it again produces sticky end or overhanging ends. So this is all for today. This is how the restriction enzymes, they go for their action. And we have already studied how are they named, how was its discovery done, and all. Thank you so much.